All right. Okay, let's continue. Let's continue. Good evening. So, in the previous class, friends, we just start, I want you to be very patient, friends. It will take 1.5 hour or 1 hour, 45 minutes. It will take, I'll teach little slowly, but effectively. So, because this could be a final revision to all of you. So, have patience throughout the class and start learning. Okay. And always think that this is my last day of preparation and I have to give my best. Then only you can, you can continue throughout the class. Okay. Don't give up in the middle. All right. So if all of you quickly recall in the previous slide, as we started discussing about photolysis of water, I mean to say, uh, whenever the P700 loses its electron, or when the P700 is deficient of electron, who is donating the electron? Undoubtedly, PC is donating the electron. Similarly, friends, whenever the P680 is deficient of electron, who is donating the electron? Undoubtedly, friends, we can say that water molecule is donating, right? That is what we all studied. So we also studied very clearly in case of a non-cyclic photophosphorylation, how the NADPH is getting produced. Now we have to focus on how exactly the ATP is getting synthesized. I have already told to all the students very clearly the production of ATP in case of a light reactions, friends, it is exclusively facilitated by a proton motive force. I'm going to ask you many questions right now. I want all of you has to think properly and answer. I believe you all read the concept multiple times, by that you never make any mistake. Now listen. So every student understand. First, I'm going to explain you how exactly the ATP synthesis occurs. What is this? You can observe the picture very clearly. This is the thylakoid membrane. You can see photosystem 1, you can see photosystem 2. You can see a lot of electron accept, not audible. I think I am audible to everyone. Just confirm me, everyone. Am I audible to everyone? Quickly. Shafia, just refresh yourself. Refresh. Why don't you refresh? All right, all right, all right. Please continue. Yeah. So what I'm trying to explain you. Yeah. So proton motive force is the one which is actually involved in the synthesis of ATP. Focus on it. So you can see it, right? This is the P680, cytochrome B6F, plastoquinone, P700. Okay. Now all the students very carefully understand this concept. It is very tricky one. Now, as I told you that ATP synthesis is exclusively depends on the proton motive force. So first we all has to understand from where these protons are coming from, or from where these protons are releasing from. That is a very crucial point we have to focus on. So friends, from the P680 just now we studied, right? Who is the electron acceptor from the P680? Just now we studied pheophytin. Pheophytin is the electron acceptor from P680, and from the P680, from the pheophytin, the electron will be transferred to the plastoquinone. Now, plastoquinone to cytochrome B6F, cytochrome B6F to plastocyanin. Now, all the students follow me very correctly without having any confusion. Now, friends, what the pheophytin will do? Pheophytin will donate the electron to the plastoquinone. Then you know what happens? Plastoquinone, upon accepting the electron from pheophytin, plastoquinone getting reduced to the PQH2. Now, very interesting question. Interesting point. If the plastoquinone want to get reduced to the PQH2, electron is not sufficient. It requires the proton also. So by using both the proton and electron only, plastoquinone is getting reduced to the PQH2. Who donated electron? Pheophytin donated. Then plastoquinone has to take the proton from somewhere. So from where it is taking the proton? Undoubtedly, it is taking the proton from stroma. Then plastoquinone reduced to the PQH2. Don't get confused with that one. Now, all the students try to understand. Now, do you really think the PQH2, which is a reduced form, do you really think it will keep the electron with it? Never ever. It never keep the electron with it. So it is sending the electron to the cytochrome B6F. Okay, then upon donating the electron to the cytochrome B6F, friends, PQH2 oxidized back to PQ. We got that point. There's no doubt. Now, electron donated to cytochrome B6F. There is no doubt. But what about the proton? So you can see the picture. All the protons are releasing into the lumen. You can see that. I know most of the people did not understand this. Follow me very correctly. I am explaining to all of you how the ATP is getting synthesized. ATP synthesized in the non-cyclic photophosphorylation by a force called as proton motive force. So how the protons are releasing, I am explaining to all of you. Whenever P680 donate its electron to the pheophytin, from the pheophytin, electrons donated to the plastoquinone. Plastoquinone, upon accepting the electron from pheophytin, upon accepting the proton from the stroma, it got reduced to the PQH2. Now, PQH2, which is a reduced form, it never keep the electron with it. It is sending the electron to cytochrome B6F. And what about the proton? Look into this. It is releasing the proton into the lumen. Now, you tell me. So, all the time this reaction is continuously happening. So, friends, the protons are releasing into the lumen continuously. At the same time, in the previous slide only we studied, whenever the water molecule break down, what's happening? There is a release of proton also. So friends, protons are coming from PQH2 also. At the same time, when the water molecule break down, there also protons are releasing. Now, what's the point? Now, the point is that the concentration of protons become extremely high. Every student has to understand me. Now, the concentration of protons become extremely higher in the lumen when compared to the stroma. When compared to the stroma, lumen having the more proton. Now, you know what happens? To maintain the charge balance, every student understand. To maintain the charge balance, all the protons starts moving from where to where? All the protons starts moving from lumen to the stroma. How they are moving? You only look into this. Proton always move in the form of a wave, like this. So whenever the proton is moving in the form of a wave, all the students has to understand me. Here, it generate energy. I'm using the word generate, okay? It generate the energy. So friends, who will use that energy? That energy can be used by ADP and PI, and it can give rise to the ATP. Who will catalyze this reaction? ATP synthase enzyme, which is also called as F0F1 complex. F0F1 complex is going to synthesize that ATP by using the energy generated from the proton. This is how ATP is getting synthesized. Most of the times, they used to ask the question directly from here, whatever we discussed. But last time, they asked a question which is it's not a tricky at all. It's informative based. Every student listen to me very, very carefully. So friends, I strongly believe you remember there are two type of plastoquinones are there. One is called plastoquinone A and plastoquinone B. Tell me all the students, among the plastoquinone A and B, together what do we call them as? We call them as primary quinones. This is a very interesting point. We call them as primary quinones. Example, plastoquinone A and B. My question is, among the quinone A and B, which is a tightly bound, which is a loosely bound? I want all the students has to answer this. Which is a tightly bounded, which is a loosely bounded? All the students remember, every one of you. Plastoquinone A will be tightly bounded. Plastoquinone B will be loosely bounded. Now you tell me, if I am using the word tightly bounded or loosely bounded, all the students answer. Among PQA and PQB, which can act like a mobile carrier? All the students answer my question. Among the A and B, which can act like a mobile carrier? Undoubtedly, plastoquinone B can act like a mobile carrier. So all the students has to understand my statements very correctly. So both undergo the reduction, right? See, friends, I hope you're getting my point. When the pheophytin donate the electron, plastoquinone A also undergo the reduction, plastoquinone B also undergo the reduction. But both reduction types are different. Every student has to remember, friends, plastoquinone A, it is getting reduced to plastosemiquinone. You have to remember this compulsory. If you forgot this, you can't answer. Plastoquinone A is a tightly bounded. Upon accepting the electron, it is getting reduced to plastosemiquinone. And what about the QB? QB is a loosely bounded mobile carrier and it is getting reduced to plastoquinol. This is a very important statement. All the students has to remember compulsory for the exam. Never get confused. A is tightly bound, B is loosely bound. A get reduced to plastosemiquinone, B is getting reduced to plastoquinol. Never get confused. Now every student try to answer the question. I only given the answer, but you recall it quickly. Don't make it wrong, okay? They're asking correct. Take your time, take your time. Don't give random answers. Come on, be active. Three. Okay. Anyone else give me the answer? What about others? All right, all right. Okay. Look into the question. The first electron released from the reaction center P680 is transferred to the plastoquinone A, which is reduced to the plastosemoquinone. Absolutely true. Option B says that QA is the mobile plastoquinone. That is a wrong statement, because QA is the tightly attached one. The first electron transferred from QA to QB converts the QB into plastosemiquinone. That is wrong, right? Because QA is going to reduce it to plastosemiquinone. So option C is also wrong statement. Option 4 says that QB is tightly bounded. No, QB is a loosely bounded. So the only statement that is correct is option A. Hence the correct answer is option 3. I think many people has given the wrong answer for this question in the exam. Okay, slowly think and analyze. All right, all right. Okay, so now we have very clearly understand the differences between the cyclic and non-cyclic. Now look into the picture. Very simple. You already studied it. In case of a cyclic photophosphorylation, we studied it is important. There is only one photosystem. In case of a non-cyclic, both photosystem 1 and 2 both are involved. Very important. Here electron travels in a cyclic manner, electron travels in a non-cyclic. The significant point, breakdown of water only takes place in the non-cyclic, it never takes place in the cyclic. As we already studied, both ATP and NADPH cannot synthesize. Only ATP, there is no NADPH, but here both we can synthesize. Again, I have told to all of you many times, friends, in our exams, which is most common, the most common is the inhibitors or the regulatory questions. So that's why you have to focus on remembering the inhibitors compulsory for the exam. Never neglect it. So there are whatever I have mentioned in the slides, they are very oftenly asked example, but beyond that there is no possibility that they'll raise the question. Maybe in the electron transport chain of mitochondria, they may raise more questions. But in case of photosynthesis or thylakoid membrane, these three are enough. So there are many herbicide that block the electron flow. What are those? DCMU, triazine, paraquat. These three are said to be what, friends? Inhibitors. Suppose, look into the picture correctly. DCMU, also called as diuron. All the students look into this. This is the P680 and this is the P700. So what I can say to all of you is DCMU, all the students understand me what I'm talking. DCMU is exactly blocking the electron flow. What, friends? DCMU is exactly blocking the electron flow from P680 to P700. And more specifically, if you ask me, it is exactly blocking the binding site between the QA and QB. In general, you can use the word, it is blocking the electron flow from P680 to P700. More specifically, it is blocking the electron between QA to QB. You have to remember that. Then paraquat, which we also call it as methyl vialogen. Here, I can tell you in a simple words, paraquat is involved in inhibiting the electron flow that is involved in the production of NADPH. What I can give you a statement? I can give you a statement that methyl vialogen or paraquat, it is blocking the electron flow that is involved in the production of NADPH. That's the simple point I want you to remember. Last time I have already solved the question. I am not solving again, but I am requesting every one of you remember the inhibitors compulsory. Okay, we will look into the dark reactions. Then we will come to the photoprotective reactions. We will focus on the dark reactions first, because which is more important for the exam. Okay, now listen. Right now, what we studied is the light reaction. What is the ultimate goal of light reaction? Production of energy. Okay, successfully plant produced the energy. Now, but production of energy is not the ultimate goal for the plant body. What is the ultimate goal for the plant body? Ultimate goal for the plant body is reduction of the carbon into glucose. This is the significant goal of the plant. So for that, it produced energy. Now, by using this energy, it is reducing the carbon into glucose. So whenever it is strictly dependent on the energy, what is the point of light? There's no requirement of light. Whether the light is present, whether the light is absent, doesn't matter. Plant reduce the carbon compulsory easily to the glucose by using energy. That's why we can call the dark reaction or light independent reaction. Ultimate goal is what? We are fixing the carbon into glucose, right? That's why we can call carbon fixation. And another point, listen every one of you, you also call carbon assimilation. What is the why we are using the word carbon assimilation? Because friends, undoubtedly the plant is going to assimilate the carbon into its body. Why? In order to break down into glucose. That's why we also call it as a carbon assimilation reactions. So based on the dark reaction or based on the carbon fixation reaction, I told to all the students multiple times, all the plants present on this planet Earth, broadly categorized into C3 plants, C4 plants, CAM plants. Based on what? Based on the carbon fixation. Again, I'm just reminding you quickly, 95 to 97% of the plants present on this planet Earth, they belongs to the C3 pathway only. And what about the C4? Mostly the monocots. I can say simply more than 18,000 of angiosperm families belongs to the C4 pathway. And what about the CAM? Everyone commonly know that the plants which are living in the desert or the plant which belongs to the Crassulaceae family. They belongs to the CAM. And based on what we actually categorized, based on the temperature condition, at the same time based on the carbon fixation also. Now let's focus on the one by one plant clearly. Now, I'm asking one common point, one simple statement I'm asking you. Every student say yes or no the statement. C3 plant contain more rubisco, listen my statements very correctly. C3 plants contain more percentage of the rubisco compared to the C4 and CAM. Again, all the students listen my statements correctly. C3 plant, listen, you are not listening my statements. You are not listening. Listen to me. C3 plant contain more percentage of rubisco compared to the C4 and CAM. Every student remember my words. Rubisco is present in every single plant at the same percentage. The percentage never be changed. It will be same in all the plants, but their activity will be differ. All the students understand the statement correctly. Only activity will be differ, friends. Their percentage will be same. I am not talking about the abundancy. I am only talking about the percentages. I am not at all talking about any kind of abundances. Now, let's talk one by one. Now, friends, one point is crystal clear. Whether it is a C3 plant, whether it is a C4 plant, whether it is a CAM plant, what's the point you have to understand? Plants are basically using the ATP and NADPH. Look into this. By using the ATP and NADPH, plant is going to reduce the carbon into what? They are going to reduce the carbon into the glucose molecule. Very easy. But friends, what's the interesting point you all people can see in this picture? None of the plant never reduce the carbon directly into glucose. You can see that, right? First, the plant reducing the carbon into 3 carbon containing compound. That's a very crucial point. After that, only the 3 carbon containing compound can be converted into the 6 carbon containing compounds such as the glucose. Now, we all people studied very clearly that all the light reactions are exclusively taking place in the thylakoid membrane. Why? Because all the photosystems, all the electron acceptors are present over there. Similarly, where does the dark reactions will take place? On the stroma. What's the reason? Why it has to take place only in the stroma? The reason is very simple, because all the enzymes such as most important enzyme rubisco, another important enzyme pepper carboxylase, and many more, they are all present in the stroma only. That's why dark reactions are exclusively taking place in the stroma. Now let's talk the first and most important one, that is the C3 pathway. Okay, before that, every one of you quickly solve the question. I forgot it. Quickly. I forgot it, but it's important question. Everyone. What about others? No, what happened? All right. Okay. So you can see it, right? Electrons from P680 moves first to quinone. That is wrong, right? From the P680, it comes to the pheophytin to the plastoquinone. Is it? So we can say that the option A itself wrong option. So hence easily you can come to the conclusion option D according to the options. P700 can receive the electron from PC. NADPH is produced at the end of light reaction. Hydrogen ions, we discussed, right? The protons can be pumped in the lumen. That is also true. Hence, the correct answer will be what? Obviously, option D. Very good. Now, all the students understand. Now, again, all the students use your brain slowly and answer. You have to think properly. Think properly. Can I use a statement that CAM pathway, CAM plants and C4 plants also perform the C3 pathway? You have to think properly and answer. CAM plants and C4 plants also performs the C3 pathway. Of course, true. The point is that in every single plant, C3 pathway is the most common pathway, because plants are going to fix the carbon by using C3 only. But in a C4 and CAM, it occurs in a different way. All of you focus. So the Calvin cycle or C3 pathway occurs in three stages. One is called fixation, second one is called reduction, and the third one is called as a regeneration. No need to get confused, friends. You have to remember compulsory. So we are fixing the carbon into 3-PZA, then we are reducing the 3-PZA into G3P, then finally after production of glucose, we are regenerating. Anyway, I'll explain step by step properly. Follow me very quickly. And I am requesting all of you, no need to remember any kind of a structure, but majorly focus on the calculation part. Calculation only very crucial here. Okay. Okay. Now quickly answer. What is our ultimate goal? All the students focus on the diagram very correctly without having confusion. What's our ultimate goal? Our ultimate goal is fixing the carbon into what? Fixing the carbon into glucose. That is the ultimate goal of a plant body. So now the question is very clear. If the plant, I'm using the word plant, if the plant want to fix the carbon into half glucose, every student remember, if C3 plant want to produce half glucose molecule, the C3 cycle has to turn 3 times. This is a crucial step every student has to remember. So if a C3 plant wants to produce a 1 glucose molecule, the C3 cycle has to turn 6 times compulsory. There's no need to get confused here. Now, I am just using a word that we have to fix the carbon into glucose. Whenever I want to fix the carbon into glucose, someone has to accept that carbon. Remember everyone, someone has to accept that carbon and they have to convert that particular carbon into glucose. So who is accepting that carbon? That carbon can be accepted by a carbon acceptor, which we generally call it as the RUBP. Again, people are getting confused between the RUBP and Rubisco. All the students keep the point in your mind. Rubisco is the enzyme. RUBP is the carbon acceptor. Never ever get confused with that. I'm explaining you directly, friends. I always talk about half glucose. By that, we can calculate for multiple glucoses easily. So, in order to produce half glucose, cycle has to turn 3 times, which means one time one RUBP will come into the picture. Then 3 times, how many RUBP will come into the picture? Undoubtedly, 3 RUBP comes into the picture. Everyone can look into the picture. One RUBP is 5 carbon. 1, 2, 3, 4, 5 carbon. So 3 RUBP, each one is how many carbon? 5 carbon. 3 into 5, 15 carbons. And 1 RUBP always accept 1 carbon. 3 RUBP accept how many carbon? 3 carbon. So 15 carbon plus 3 carbon will become how much? 18 carbons. Again, some people are having a confusion whenever I am explaining you 3 molecules at a time. So I will explain for 1 molecule. Don't worry about it. Focus. I want to fix carbon into glucose. For that, someone has to accept the carbon. Who can accept the carbon? RUBP. RUBP is how many carbon? 1 RUBP I'm talking about. 1 RUBP is a 5 carbon. And 1 RUBP accept 1 carbon. 5 carbon plus 1 carbon, it will become 6 carbon. We got the point. But that 6 carbon containing compound, it is completely unstable. It is unstable. So unstable cannot participate in any reaction. So that unstable, we have to break it compulsory. How can we break? We have a 6 carbons. We are breaking into 2 compounds. Each compound contain how many carbon? 3 carbon. Each compound contain how many carbon? 3 carbon. So the stable product or the intermediary product, which is a 3 carbon, that's why the name of the pathway given as a C3 pathway. No need to get confused with that particular statement. Quickly understand all the students. So I want to fix carbon into glucose. For that, I require an acceptor who is called as RUBP. RUBP is a 5 carbon accept 1 carbon from the atmosphere. 5 plus 1 will become 6 carbon. That 6 carbon break down into 2 3 carbon containing compounds. This is easy. But what is our goal? If the plant want to produce a half glucose, only half, I'm always talking about half. For a half glucose, it has to turn 3 times. 3 cycles are required. So 1 cycle 1 RUBP, for 3 cycles 3 RUBP. So 3 molecules of RUBP combined with 3 molecules of carbon dioxide. So 15 plus 3, 18 carbon. Again, we discussed that it is completely unstable compound. So that unstable compound has to break down. What is our rule? Our rule is it has to break down into only 3 carbon. It should not break down more than the 3 carbon. Now, how many carbons we have? We have 15 carbons here. We have 3 carbons here. Total, we have 18 carbons. But we have to break down into only 3 carbon containing compound. So by 3, then how many we get? We get a total 6 molecules of 3-PZA. This is called as first step. What is the first step? Carbon fixation. Just 1 minute, the screen will come back. So simple words, remember, 3 molecules of RUBP, which is a total 15 carbons, combined with 3 carbon dioxide in the atmosphere, total of 18 carbon. And that 18 carbon, all of you remember, that total 18 carbon get split into 6 molecules of 3-PZA, because our rule is that our intermediate molecule should be 3 carbon only. You got the point very correctly. That is called carbon fixation. But our goal is to fix the carbon. No, right? Our goal is to reduce. Now listen everyone. The 6 molecules of 3-PZA, 6 molecule of 3-PZA, each one is how many carbon? All the students understand. Each 3-PZA is how many carbon? 1, 2, 3 carbon. So 6 molecule of 3-PZA, each one is 3 carbon, is getting reduced to the G3P. Now the question is very clear. What is the difference between the 3-PZA and G3P? What is the difference between phosphoglyceraldehyde and glyceraldehyde 3-phosphate? Friends, both are same. There's no difference. Then, because this is also 3 carbon, this is also 3 carbon. The difference is that both are acting like a reducing form. So 3-PZA is getting reduced to into its other form called as G3P. So, but the reduction won't happen that much easily, right? If 1 molecule of 3-PZA want to reduce it to 1 molecule of G3P, we require 1 ATP and 1 NADPH. Every student understand me correctly. If 1 molecule of 3-PZA want to reduce it to 1 molecule of G3P, we have to spend 1 ATP and 1 NADPH. But what is the question here? We have 6 molecule of 3-PZA. So I have to reduce the 6 molecule of 3-PZA compulsory into 6 molecules of G3P. Then I have to spend 6 ATP here and 6 NADPH. This is called as reduction step. They can ask a question directly in the exam. In which of the step of the C3 pathway highest form of energy is consumed? Highest form of energy is consumed in which step? Which is the reduction. You have to think properly. Then all the students remember, this is the most crucial step. Now tell me, finally, how many G3P we produced? 6 molecule of G3P we produced. Just a 6. So each G3P is how many carbon, friends? You know very well. Each G3P is a 3 carbon. You can see that. Now what's happening? Out of 6 molecules of G3P, there is only 1 molecule of G3P comes out of the cycle. 1 is enough. 1 is enough. Out of 6 molecule of G3P, 1 molecule of G3P comes out and it is going to produce only half molecule of glucose. And how many G3P are left out? 5 molecules of G3P are left out. Each G3P is how many carbon? 1, 2, 3. 3 carbon. So 5 into 3, how many carbons we are having? 15 carbon. Now the point is clear, right? By using 5 molecules of G3P, ORR, 15 carbon. How many RUBP? Every student remember, how many RUBP we can regenerate? Undoubtedly, we can regenerate 3 molecules of RUBP, because each RUBP is 5 carbon. In our hand, we have 15 carbons. So I can easily regenerate 3 molecules of RUBP. But again, regeneration is also not easy step. Regeneration also we have to spend energy. So I have spent 3 molecules of ATP here as well. So I'm summarizing it in a simple manner, and I am only talking about half glucose. In order to produce the half glucose in the Calvin cycle, the cycle has to turn 3 times. So if it is turning 3 times, someone has to accept the carbon every time. Who is the acceptor of carbon? RUBP. 3 molecule of RUBP, each RUBP 5 carbon, together 15 carbon, combining with another 3 carbon, total 18 carbon, break down into 6 molecule of 3-PZA, each one is a 3 carbon. This is the carbon fixation. Now, the 6 molecule of 3-PZA is getting reduced to the 6 molecules of G3P by spending 6 molecule of ATP by 6 molecule of NADPH. Out of 6 molecule of G3P, 1 molecule of G3P comes out of the cycle and produce half glucose. Remaining 5 molecules of G3P is getting regenerated into 3 molecules of RUBP by spending 3 ATP again. So I will ask you very simple questions only. Don't be hurry, okay? Don't be hurry. First listen. First listen calmly and answer. My first question. You have to think properly, okay? I'm not making you confused. I'm just making you expert. In order to produce 3 sucrose molecules, every student think properly and answer, okay? In order to produce 3 sucrose molecules, how many number of Calvin cycles are required? That is the first question. And the second question is that in order to produce 1 sucrose molecule, how many ATP, how many NADPH are required? That's my simple question. Okay. Before this question, first answer this question. First answer third question. How many number of ATP? Simple question only. How many number of carbon dioxide? How many number of NADPH required to produce 1 glucose molecule? I am asking you a simple question. First answer me third question. How many number of ATP, carbon dioxide, NADPH are required to produce 1 glucose molecule? Every student, every one of you has to answer. Don't wait for others' answer. Ritika, what happened? You forgot everything, huh? Swati, look into that correctly. Everyone forgot, huh? What happened? Everyone forgot, huh? Most of the people. Look into this. I have asked all of you for 1 glucose. So you only look into this. Here we calculated for half glucose, right? For half glucose, you see this? For half glucose, I used 6 ATP here, 3 ATP here. So 6 plus 3, 9 ATP and 6 NADH, just for half glucose. But I have asked all the students for 1 glucose. So 1 glucose, we require 18 ATP and we require 12 NADPH. For half glucose, we require 3 carbon dioxide. Then for 1 glucose, how many we require? We require 6 carbon dioxide. Correct, na? Right. So that would be the answer for the question. What is that? 18 ATP, 6 carbon dioxide, 12 NADPH. Now answer for question number 1 and 2. Now answer for question number 1 and 2. What is the question number 1? How many Calvin cycles are required to produce 3 sucroses? Yeah. Okay. All the students answer my question. Every student answer. In order to produce 1 glucose, every student answer. Okay, let's consider. Wonderful, wonderful, wonderful. Answer, all the students answer. In order to produce 6 glucoses, how many number of times the Calvin cycle has to turn? In order to produce 6 glucoses, how many number of times the Calvin cycle has to turn? Everyone, undoubtedly 36, right? Then that is what I have asked. You all know very well, 2 glucose molecule come close together, they make 1 sucrose. Correct, na? So if they asked a question for 1 sucrose molecule, how many times Calvin cycle will be occurred is 12. Correct, na? I have asked for 3 sucroses, it is 36. Okay. Actually, they never ever ask in that way. They never mention the sucrose. Maybe in the calculations they may ask. Now tell me, for 1 sucrose, tell me friends, how many ATP and ADPH? For 1 sucrose, 1 sucrose. All the students answer. For 1 sucrose, how many ATP and how many NADPH? All of you. For 1 sucrose, how many ATP and how many NADPH? What is there to think? Very good. So for 1 glucose, we require 18. So 1 sucrose means 2 glucose, right? Then we require 36 ATP and here 24. That's all. What's there to get confused? Good, good, good. Perfect, perfect, perfect. Right, right. Okay, don't get confused because that kind of questions they use it to ask very commonly. Okay. Now, so in the calculation, what you people can observe, 9 ATP are converted to 9 ADP. And 6 NADPH are converted to 6 NADP plus. So always remember, for 1 glucose molecule, how many number they may ask, you people can answer easily. To make 1 molecule of glucose, we require 6 carbon dioxide, 18 ATP molecule, and 12 NADPH molecule. Don't get confused. Okay. Now everyone answer this question. Again, this is the part B question. Quickly. You're asking consumed, okay? Correct, na? So there are only 2 steps we have spent it, right? We spent it during the reduction also, friends, as well as we also spent it during the time of regeneration also. Is it? Of course, yes. Wonderful, wonderful. Okay, again, so all the students remember, this is also very crucial, friends. I want you to understand here. Now, all the students focus, all the students focus. Out of 6 molecules of G3P, we studied that there is only 1 molecule of G3P comes out of the cycle and produce half glucose. That is true. But friends, what's the point? What is the fate of this particular glucose? Every student commonly know very well, in case of a plant, the sugars can be continuously transported through the phloem to all parts of the body. All parts of the plant body. But you know very well, in the phloem, glucose cannot be transported. Only there is a one saccharide, that is the disaccharide sucrose only can be transported. So which means that glucose, once it is synthesized by the Calvin cycle, it has to be converted into the sucrose to convert into a utilizable form in the plant. So how it can be converted? Again, by using certain enzymes. I don't know whether you people observed this or not. In the 2023 afternoon shift, the question paper, the question is from this slide. Okay. So even I did not expect that the question is exactly from this slide. Okay. This we will include in the PPP pathway. There I will explain to you. This is the question they have asked. So glucose can be converted into the sucrose where, friends, in the cytosol. That is also very important location. And glucose is getting converted into the starch by in the chloroplast stroma. That is very important, friends. You can see the location. Cytosol as well as the stroma. There are two different locations are involved in synthesizing it. And these reactions are absolutely easy reactions. There is no point of feeling difficulty also. But I am requesting every student, because nothing to explain here. I want all of you has to remember this equations compulsory. Equations in the sense, suppose example, this is a triose phosphate, which is what? A 3-carbon containing compound, right? Now that 3-carbon containing compound combined with another 3-carbon, it is given rise to the fructose 1,6 bisphosphate. That fructose 1,6 bisphosphate treated by a bisphosphatase enzyme cleaved the phosphate group, converted into fructose 6-phosphate. Fructose 6-phosphate convert into its isomer glucose 6-phosphate. Glucose 6-phosphate, there is a change in the position of the phosphate group. It is converted into glucose 1-phosphate. Glucose 1-phosphate converted into UDP glucose. I have told to all the students multiple times, glucose always depends on the UDP when compared to the ADP and ATP. Now that UDP glucose, this is only one glucose, right? That UDP glucose combined with another UDP glucose in the presence of a sucrose synthase enzyme, it is converted into what? Sucrose. I don't want you to remember the reactions. It's not required. What I want you to remember is the enzymes involved in the sucrose synthesis, the enzyme involved in the starch synthesis. One is called sucrose phosphate synthase. Another one is starch synthase. Starch synthase present in the chloroplast. Sucrose phosphate synthase is present in the cytosol. I am requesting you not to neglect, okay? Because if you don't revise at the end, you will forget faster. So don't neglect it under any circumstances. This is what a simple C3 cycle that happens in every plant. But here, the C3 cycle is not important. What is very important for the exam point of view is the enzyme, the versatile enzyme, that is the Rubisco. What is the speciality of the Rubisco? See, this is an enzyme, right? This is an enzyme called Rubisco. What's the function of an enzyme? Every one of you know that the enzyme function is converting the substrate to the product. That is the ultimate function of every enzyme. Now the question is here, does one enzyme can only convert one similar substrate into product, or the enzyme can convert different substrates which all of a different categories convert into product? Never. Because from the 11th standard we all studied, enzymes are specific in nature. What is the definition? Enzymes are specific in nature, which means, friends, one enzyme can convert one specific group of substrates into products. It cannot convert all. But if you look into the speciality of a Rubisco enzyme, this is the Rubisco, right? And this is what we generally call active site of the enzyme. But what is the speciality of a Rubisco here? Rubisco enzyme is involved in converting the gases into products. What gases it can convert into product? CO2 it can convert, and O2 it can convert. So now you have to remember a point. Rubisco is the enzyme. It is converting the substrates of same category. Both are gases. So the enzyme converting the gases into product. But the question is that, does the enzyme can have a separate active site for carbon dioxide and separate active site for oxygen? Never. There is only one active site. In this active site only carbon dioxide has to come and oxygen has to come. If carbon dioxide is dominating, oxygen cannot fix. If oxygen is dominating, carbon dioxide cannot fix. So here enzyme is not doing anything. Gases are fighting among themselves in order to associate with the enzyme. How is that possible? It is exclusively depends on the environment. So environment is deciding whether the carbon dioxide has to be fixed by the enzyme or the oxygen has to be fixed by the enzyme. Who will decide? The environment, not the enzyme. Because the enzyme function is what? If a substrate comes and attaches, it just convert into product. So which substrate has to come, that is decided by the environment. That's a good point. Now, in every single plant, C3 pathway is happening. And very important point, The plants which are belongs to the C3 pathway, they compulsory, I'm using the word compulsory, you know that? I'm using the word compulsory. They will show the C2 pathway also. It is majorly because of environmental conditions such as temperature. Now listen. Why did I use the word expensive reaction? Why did I use the word waste reaction? Listen, all the students understand. This is a crucial one. Our ultimate goal of the enzyme is to fix carbon into glucose, because plant has to reduce only carbon into glucose. Why the plant will reduce the oxygen into glucose? That is not even possible. First of all, no plant cannot reduce the oxygen into glucose. That is impossible. Plant has to reduce only carbon into glucose. But unfortunately, carbon dioxide and oxygen is competing with this enzyme. Then you can't fix the glucose. How to solve this problem? Before solving the problem, first let's understand why this problem is encountering, why the plant is facing this problem. Now listen, whenever the enzyme Rubisco fix oxygen instead of the carbon dioxide, that reaction we call it as photorespiration or expensive side reaction. I'll tell you why did I use the word expensive side reaction. I can use the word side reaction. Now, why did I use the word expensive? Because we are spending a lot of energy here unnecessarily. Just now you studied plant struggled a lot to produce ATP and NADPH in the light reactions. But when it comes to the photorespiration, we are spending all these unnecessarily. That's why we called expensive side reaction. Now you can ask me, sir, you said that there is only one active site is there. So when the carbon dioxide when the carbon dioxide comes, the enzyme is fixing the carbon. Then how the enzyme can also fix the oxygen? So you have to understand the Rubisco is a versatile enzyme. The speciality is it is having a carboxylase activity also, oxygenase activity also. So it is having the ability or capability to fix anything. So whichever comes into the picture, it can fix it. So which has to come depends on the temperature. Now you see this. Let's consider a condition, high temperature. Every student very commonly know that if I ask you oxygen and carbon dioxide, which can solubilize very faster at higher temperature, undoubtedly without thinking you all people can say that carbon dioxide. Carbon dioxide having the highest solubility. But oxygen also having the solubility, there's no doubt about it. But compared to the carbon dioxide, oxygen solubility is little low. Let's consider oxygen and carbon dioxide, both are present at 100%. Both are present at 100%. Now the temperature is, let's consider the temperature is 45 degrees. At 45 degrees, what's happening? Out of 100% of the carbon dioxide, 90% of the carbon dioxide get dissolved, completely solubilized. You know how much amount of the oxygen gets solubilized? Hardly 50%. How much carbon dioxide left out? Only 10% is left out. How much oxygen is left out? 50% is left out. Then undoubtedly oxygen will dominate the carbon dioxide in the active site of the Rubisco. Then Rubisco successfully fix only the oxygen. So you remember, at a high temperature, the solubility of oxygen in the cytosol and chloroplast stroma is less affected compared to CO2. Why? Because the CO2 having a highest solubility rate, which leads to the photorespiratory condition. Never get confused with that. So look into this, because in the given exam, they asked a statement, O2 and oxygen, carbon dioxide having a different active sites in the Rubisco. That is a wrong statement, friends. Oxygen and carbon dioxide compete for the same site on the Rubisco. That's a very important point. Now all the students keep on answering my questions. This is a very important statement. If you understand this, the pathway is very clear. Okay. The people who are attending only first time, you may feel little difficulty. Okay. If you have never read the concept. Now listen. So first RUBP, let's take which is a 5 carbon. combined with one carbon dioxide in the atmosphere, there is an enzyme Rubisco. Now here Rubisco is fixing what? Only carbon dioxide. Then how many intermediates we will get? All the students quickly answer my question. When RUBP combined with one carbon in the presence of Rubisco, where the Rubisco is only fixing the carbon, how many intermediate molecule we get? Maria, you forgot. Everyone forgot. Wow, everyone answer. We get only, what happens to others? How many 3PZA we get? How many intermediates we get? Undoubtedly, we get only 2 intermediate molecule. What is there in that? I have told to all of you, our intermediate should be only 3 carbon. So this is a 5 carbon plus 1 carbon, 6 carbon. In the presence of Rubisco, how many intermediate I will get it? Undoubtedly, I will get only 2 molecules of 3PZA only. There's no doubt about it. Correct, na? Very good. Now, same thing I am remodifying it. Same thing I am remodifying here. Same RUBP, every one of you, combined with the oxygen now. There's no carbon dioxide. Same Rubisco enzyme. My question is crystal clear. How many molecules of intermediates we get it? Intermediate means, my friends, intermediate should be always 3 carbon. Intermediate should be always 3 carbon. Now you tell me, how many intermediates we get it? Every one of you. How many intermediates we get it? Undoubtedly, we are going to get only one intermediate. That is one molecule of 3PZA only. How many carbons are left out? Every one of you quickly. How many carbons are left out? All of you. How many carbons are left out? 2 carbons are left out. Yes or no? 2 carbons are left out. My friends, since so one 3PZ and one 2 carbon containing compound. You know what do we call this 2 carbon containing compound? We can call phosphoglycolate. Okay. Now my question to all the students is very simple question. My question is very clear. My question to all the students, whenever RUBP fixed the oxygen, sorry, when the Rubisco fixed the oxygen, my question to all of you is very clear. Glucose synthesis will be occurred or not? In other words, glucose synthesis will be continued or not? Whenever Rubisco fixed oxygen instead of the carbon dioxide, glucose synthesis will occur or not? Wonderful. There will be a no Calvin cycle. Now, what should I do now? If I want to allow the Calvin cycle to continue, what should I do now? I have to convert, all the students listen, I have to convert this 2 carbon containing compound back. Every student listen, every student listen. I have to convert this 2 carbon containing compound back to the 3PZA. Then only Calvin cycle will be continued. Then some students asked a doubt that, sir, now we are left with the 2 carbon. When we add one extra carbon here, then we can easily produce the another molecule of 3PCA. Your doubt is a good doubt only. But do you really think we can just add the carbon and we can convert it that much easily? No. You have to move into every single compartment. In order to convert this 2 carbon into another 3 carbon, you have to roam every compartment and you have to spend a lot of energy. That's the reason we call it as expensive side reaction or the waste reaction. Why did we use the word C3 pathway? Because the stable or intermediary product is 3 carbon. Why did I use the word C2 pathway? Here, I don't use the word stable product. Here, the end product produced is the 2 carbon in the photorespiration. That's why, friends, photorespiration is also called as what cycle? C2 cycle. The 2-phosphoglycolate is a 2 carbon, right? That's why we call it as the C2 cycle. No need to get confused. Now every student quickly look into the point. Again, the same asked question. Even though you get a one statement in your exam, that is really helpful to you. Whomever having a confusion, whomever did not understand, focus on this diagram very accurately. Okay, focus. Every student focus. This is what, friends? This is RUBP. Okay, this is RUBP. RUBP combined what? Combined with oxygen, because there is no carbon dioxide. In the presence of what? In the presence of Rubisco. Okay. What do we got? Tell me. Every student look into this. We got only one 3-PZA. You can see this very clearly. We got only one 3-PZA and what we got? We got one 2-PZA, 2-phosphoglycolate. Now, you only look into this. What I am doing now, this 2-PZA, I am bringing from chloroplast to peroxisome, peroxisome to mitochondria, again mitochondria to peroxisome, again peroxisome to chloroplast. Finally, I am converting into the another 3-phosphoglycerate. Then only I can continue the Calvin cycle. Please don't get confused with that particular point. Easy to understand, not at all difficult. Now focus, try to understand me correctly. So I have produced one molecule of 3-PZA. I have produced one molecule of 2-phosphoglycolate. How can I convert this 2-phosphoglycolate back to the 3-PZA? By the help of 3 cell organelles. We called CPM. Again, you know, right? Many times asked a question. Chloroplast, peroxisome, and mitochondria. Don't get confused. So you only look into this. This 2-phosphoglycolate, is it necessary to remember the steps? Of course, friends, because more than 3 to 4 times we got the questions from the steps also. Hence, I encourage all the students to remember the steps also mandatorily. So the 2-phosphoglycolate converted into glycolate. How is that possible? So simple. It is just losing its phosphate group. Upon losing the phosphate group, 2-phosphoglycolate convert into glycolate. Now the glycolate leave the chloroplast. This is the question they directly asked in the exam. Which is the first metabolite entered into the peroxisome? The answer is glycolate. So glycolate leave the chloroplast, entered into the peroxisome. Now what happens to this glycolate? This glycolate combined with the hydrogen peroxide, then converted into glyoxylate. Now this glyoxylate converted into glycine. Every student know very well. Glycine is the optically inactive simple amino acid. Look into this. Look into this. Now this particular glycine again from the peroxisome entered into the mitochondria. This is the most crucial step, friends. Now the glycine, listen. Till now, I have used a word that we are spending the energy unnecessarily in the photorespiration. That is the reason we call it as expensive side reaction. But you see here, glycine is converting into serine. Surprisingly, we are gaining the energy. But I have told you just now, we have to spend energy. But you see this, we gained energy here. But listen, just remember this. When the glycine converted into the serine, all the students listen. Now this glycine, when the glycine is converting into serine, NAD plus converted to NADH. So reduction happened. There is a energy got produced. And there is a release of ammonia. Every student tell me, what is the fate of this ammonia? Or where this ammonia is going to enter? Or what happens to this ammonia? I want all the students has to answer. Very good. That ammonia is entering into the nitrogen metabolism, where it is getting converted into amino acid. Now, my question to all the students is crystal clear. Tell me, Mauli, tell me, Ritika, where this particular ammonium is getting fixed into amino acid. My question is, the ammonium is getting fixed in the chloroplast, mitochondria, or peroxisome? That is my question. Where this ammonium is getting fixed. Wonderful. Very good, Swati. It is getting fixed again in the chloroplast. That's an important statement. Very, very good, Shafia, because ammonium assimilation happens in the chloroplast. Wonderful. So now glycine converted into serine. Now what happens to the serine? Serine again came to the peroxisome. Now serine converted to hydroxypyruvate. Hydroxypyruvate converted into glycerate. Now you see the picture, friends. Here reduction occur. Here again, NADH is getting oxidized back to NAD+. So oxidation is happening. My friends, I have produced energy here. I have spent energy here. Both got balanced. Now glycerate from the peroxisome entered into the chloroplast. Now you look into this. The glycerate. How the glycerate converted into 3-phosphoglycerate? How is it possible? We have to add the phosphate group, right? How can I add the phosphate group? Someone has to donate the energy. Who is donating the energy? Undoubtedly ATP. So I will quickly recap this. Follow me, everyone. So whenever the Rubisco fixes the oxygen, we get 1,3-phosphoglycerate and we get 1,2-phosphoglycolate. That 2-phosphoglycolate lose the phosphate group converted into glycolate, and that glycolate entered into the peroxisome, converted into glyoxylate. Glyoxylate convert into optically inactive amino acid glycine. From the peroxisome, glycine enter into the mitochondria. Glycine converted into serine. When the glycine converted into serine, there is a production of NADH and release of the ammonia. That ammonia again go back into the chloroplast, and it is involved in the ammonium assimilation, which we will discuss in the later. Now, serine again come to the peroxisome, converted into hydroxypyruvate. Hydroxypyruvate converted to glycerate. Again, here oxidation. So we gain energy, we spent energy, got balanced. Now, glycerate again come back to the chloroplast, and it is getting converted into 3-phosphoglycerate finally. Finally, we produce 2 molecules of 3-phosphoglycerate. Now you can ask me, sir, now what is the problem here? Your question is a very valid. Sir, C3 plant, it is showing the C2 cycle. Now what is the problem? See, friends, here you are spending the energy unnecessarily. First point. Second point, very important point, you are wasting a lot of time. So the preparation for the food, it will take long time for the plant. That's the reason plant always try to escape the C2 cycle, because this many compartments they have to move. They have to use multiple enzymes. See, if you know, right, if all the enzymes wants to work, also a lot of energy is required, and it takes a lot of time, lot of energy. That's why plants always try to escape the C2 pathway. But it is depends on what? It is exclusively depends on the temperature condition, which means it is exclusively depends on the human beings only. Say at a specific condition, I'm just telling you, you take a metropolitan cities like a Delhi, right? If you look into the Delhi during the summertime, I'm especially telling you, the temperature is of course very high. But during the normal time also, you know very well, right? Because of, releasing of several toxic chemicals into the atmosphere, the temperature is increasing continuously. Now you imagine, now, when the temperature is increasing continuously, how the plants present in the Delhi, they will always prefer their C3 pathway. They always prefer C3 pathway. It is not possible. Because of the more temperature, they keep on performing the C2 pathway. Then you know what happened? Photosynthetic efficiency of the plant will decrease. As a result, I can say that lifespan of the plant also getting decreased. That's a very important statement. And I really don't know, there is no evidence also, but many research is suggesting that most of the plant in the future, I'm using the word in the future, most of the plants, they may shift their pathway from C3 to C4, especially in the areas where the temperatures are extremely higher. But we really don't know. We really don't know. Is there any mechanism is existing in the plant that they can shift their pathway from C3 to C4? We really don't know. But the research suggests that there may be a possibility because of a excessive temperature. That is what C3 plant, very important one. But the entire planet Earth is not only occupied by the C3 plant, we have the C4 also, we have the CAM also. Is it? So, but C3 are dominating plant. But what is the speciality of C4 and CAM? These two plants are very less in number when compared to the C3 pathway. C3 plants, because 97% of all the plants are C3 only. These are all less in number. Now, what's the point? Even though they are less in number, they are having a lot of adaptations. What is the meaning of an adaptation? So they have habituated, I can say that. Instead of using the word habituation, I can say that they developed certain mechanisms. They developed certain mechanisms. They can easily escape photorespiration. I don't use the word escape. Escaping is wrong statement. They will actually prevent the photorespiration. See, escaping means C4 pathway also show the photorespiration, but that can be escaped. That is wrong. They will directly prevent it. Let's see how. And you also know, right? Multiple times the question was asked from the C4 pathway, especially from the Kranz anatomy. Now, what plants comes under the C4 pathway? There are more than 18,000 type of the angiosperms, especially monocots, belongs to the C3 pathway. I hope all the students remember my example also. Give me the example of a dicot that follow the C3 pathway, that follow the C4 pathway. Actually, monocots will prefer the C4 pathway. Give me the example of a dicot that follows it. Very good. So what was that? Amaranthus is the name of the plant. Even though it belongs to the dicot, it will follow the C4 pathway. I want all the students has to remember it compulsory. So you can remember any kind of a monocots example. But most commonly asked examples in the exam is maize, sugarcane, sorghum, millet. These four are the major plants that they used to ask very commonly. Now, if you quickly recall, why did we use the word C3 pathway? Because the stable compound is 3 carbon. And why did we use this C4 pathway? The stable compound is the 4 carbon. If you quickly recall, what is the stable compound here in the C3 plants? 3-PCA, because you have to remember them. And what will be the C4 stable compound? OAA, oxaloacetic acid. And every C4 plant having a specialized leaf structure that is called Kranz anatomy. Look into this. So this is the C4 plant. If you do the cross section of this leaf, from here also we got a direct question in the exam. If you look into the Kranz anatomy very clearly, look into this. This is the leaf. You can see this. This is called upper epidermis. This is called lower epidermis. Correct, na? And you might observe most of the stomatas are located on the lower epidermis only. All the students look into this. You can observe only there are two type of cells. One is called as mesophyll cells, and another one is called as bundle sheath cells. Okay. And friends, if you look into the picture of a mesophyll cells, you can see, right? Mesophyll cells, where they are present on the surface. Correct, na? Here also mesophyll cells are there. Here also mesophyll cells are there. But you can look into one common point. All the mesophyll cells, look into this. There is a lot of intercellular spaces that you can observe. So what is the statement they have given in the exam? All the mesophyll cells are very tightly attached with each other, which is absolutely a wrong statement. Mesophyll cells are having a lot of intercellular spaces. They're freely arranged and they're located exclusively at the surfaces. But if you look into the bundle sheath, you look into this. They're located very deeper. And can you find any kind of a intercellular spaces between the cells? No. So they are tightly associated with each other, and there's no intercellular space. But if the if the C4 pathway wants to occur, it is mandatory. We require what we require? We require mesophyll compulsory. We require compulsory bundle sheath. Without cooperation of these two, C4 pathway never occurred. But if these both wants to be cooperated, mesophyll and bundle sheath cell has to combine with each other compulsory. How is that possible? Because both can be connected with each other. Why? Plasmodesmata. This also they may raise the statement. Plasmodesmatal connections are absent in the C4 plants. which is again absolutely wrong statement. Lot of plasmodiest metal connections will be present in the C4. Okay, I'll explain the C4 little more elaboratively compared to the last time. I want all the students has to focus. Okay, because here also we have to focus on multiple calculations. Okay, especially ATP calculations. I'll teach little elaboratively. Later I will leave. Okay, be patient for 20 minutes. Understand me correctly. Our ultimate goal is what? To prevent the photorespiration. There's no doubt about it. Now all the students listen. How many cells are involved? Mesophyll is involved. Bundle sheath is also involved. Correct, na? Now, again, now I have told to all the students very clearly, Rubisco enzyme is present in every single plant. There's no doubt about it. Similarly, Rubisco enzyme is also present in the C4 plant. Now you have to ask me a question. Sir, if the Rubisco is present in the C4 plant, why can't you say Rubisco is the major enzyme? The reason is that in case of a C4 plants, Rubisco is present in very, very deeper of the cells. In other words, I can tell to all of you, see in the C4 plants, Rubisco is present only in the bundle sheath. You can see, right? Bundle sheath is very, very, very deeper. Now all the students answer my question. Whichever is deeper, who will transport very easily, friends? Very important statement. Carbon dioxide can diffuse very easily when compared to the oxygen. Here itself, I can tell you the reason. Here itself, I can summarize you. Why C4 plants avoid the photorespiration is, this is one of the major reason. Where the Rubisco is present? Within the bundle sheath. What is the function of Rubisco? Either fixing the carbon dioxide or oxygen. But the interesting point is that carbon dioxide only enter inside of the bundle sheath cell. Oxygen cannot enter. So there only you can understand that Rubisco only fixes carbon dioxide in the C4. Let's see how. Let's see how. Now, so if the Rubisco is present in the bundle sheath, who is present in the mesophyll? In the mesophyll, we have another versatile enzyme that is called PEP carboxylase enzyme. What enzyme? PEP carboxylase. So first, I will explain you, friends, overview. Later, I will explain step by step elaboratively. I am requesting you, it's not my guess, but I'm just assuming. This time we may get the question from the C4 specifically, because in the last June and December, we did not get a specific question. So that's why I want you to focus on it. Let me give you the overview first. Overview. This is the mesophyll. All right. And this is the bundle sheath, right? You look into this. Who is the first carbon acceptor? All the students answer my question. In case of a C3 plants, who is the carbon acceptor? RUBP. Every student answer. Who is the carbon acceptor in case of a C4 pathway? C4. C4. Forgot everyone. In case of a C4, I am asking, who is the carbon acceptor? Mauli, forgot everyone. What happens to all people? Most of people are giving wrong answers. I am asking acceptor, which is the PEP. PEP is how many carbon? 3 carbon, right? Phosphoenol pyruvate, which is the 3 carbon which accept 1 carbon from the atmosphere. And what is the enzyme? Phosphoenol pyruvate enzyme. PEP carboxylase enzyme, right? Very important, friends. Very important it is. PEP carboxylase enzyme. And how many, what we get? We get an intermediate which is a 4 carbon. What do we call it as? Oxaloacetic acid. Please turn off your videos. Don't make it completely turn off videos. What you are keeping. Turn off your videos. Don't turn on the videos. Don't disturb me in the middle. Please look into it properly. Maintain the class decorum all the time. Okay. Now, PEP carboxylase combined with carbon dioxide and we get the oxaloacetic acid. Now, that oxaloacetic acid, every student remember, unfortunately, oxaloacetic acid can't enter into the bundle sheath. Now, you can ask me why. Very important statement. Oxaloacetic acid do not have any active site in order to enter into the bundle sheath. That's why oxaloacetic acid converted into either malic acid or aspartic acid. Anything it can convert. But most of the time, oxaloacetic acid converted into malic acid. Oxaloacetic acid is how many carbon? 4 carbon. Malic acid is how many carbon? That is also 4 carbon. Correct, na? Now that malic acid entered into the bundle sheath. Now you can ask me, sir, how? Because malic acid having lot of active sites in the bundle sheath cell. Now tell me, malic acid is how many carbon? 4 carbon. Now within the bundle sheath cell, there is an enzyme called as malic acid decarboxylase. It will break down the malic acid into 3 carbon plus 1 carbon. What is that particular 3 carbon? So now this 3 carbon nothing but pyruvate. And who will take this 1 carbon? That is important here. Rubisco will take this 1 carbon. Rubisco take that particular 1 carbon and it is successfully convert into glucose. How many carbons are left out? 3 carbons are left out, which we can call pyruvate. Now the pyruvate 3 carbon again come back to the mesophyll cell. By spending ATP, it is converted into phosphoenol pyruvate again. This is the overview. Now let me explain you step by step elaboratively. It's very crucial. I want you to focus on. If you look into the C3 cycle, we read that fixation, reduction, regeneration. Now focus all the students. I'll explain step by step focus. In case of C4 pathway, carboxylation, breakdown, splitting, and phosphorylation. Because you can expect the question from the name of the steps also. So what's happening in the carboxylation? You only tell me. All the students look into this. Who is the first carbon acceptor? Phosphoenol pyruvate. How many carbon? 3 carbon. Combined with 1 carbon. 3 plus 1, 4 carbon. We are getting what? Oxaloacetate. We got that very correctly. And what is the major principal enzyme? This is a crucial point. PEP carboxylase enzyme. It is just added that carbon and converted into oxaloacetate. Now, oxaloacetate cannot enter into the bundle sheath. That's why it is getting disintegrated into either malate and aspartate. Now, you can ask me, sir, how does the oxaloacetate converted into aspartate and malate? Very important, friends. By the enzymes transaminase especially. By the enzyme transaminase and malate dehydrogenase. They converted into aspartate and malate. Now all the students follow me. Now this particular aspartate cannot enter into the bundle sheath cell. So who can enter? Malate can enter. Malate is how many carbon? 4 carbon. So malate successfully enter into the bundle sheath. In the bundle sheath, we have a versatile enzyme. What was that enzyme? Malate decarboxylase enzyme. What that enzyme is doing? That enzyme convert the malate into 3 carbon plus 1 carbon. What is that 3 carbon? What is that 3 carbon? That 3 carbon only pyruvate, and there is a leftover of 1 carbon dioxide. Who will take this 1 carbon dioxide? This 1 carbon dioxide taken by the Rubisco and fix the glucose. Now what happens to this particular 3 carbon, friends? So you can see this. The pyruvate molecules are moved to the chloroplast of the mesophyll. In the presence of ATP, it is getting converted back into phosphoenol pyruvate. Again, all the students has to remember the enzyme. The enzyme which is converting the pyruvate to the phosphoenol pyruvate. The enzyme is pyruvate phosphokinase. Don't get confused with this point. Finally, we can synthesize the phosphoenol pyruvate. Please don't get confused with this particular point. Now, without having a confusion, I am telling you the point here itself. I hope you all people remember. Here what happens? This carbon dioxide taken by whom? This carbon dioxide taken by Rubisco. Remember. This carbon dioxide taken by the Rubisco, and Rubisco continue the Calvin cycle. Every student know, right? In the Calvin cycle, how many ATP we spent? 18 ATP we spent, 12 NADH we spent. We got it. Now all the students answer my question. How many number of times the C4 cycle also turns? That's also a question, right? How many number of times the C4 cycle will turn? Everyone. How many number of times the C4 cycle will turn? See, for what? Same like C3 only. In order to produce the half glucose 3 times, in order to produce 1 glucose 6 times, there's no doubt about it. Everything is same. Now all the students has to understand me very correctly. All the students listen my statements very correctly. Whenever the pyruvate is converting into phosphoenol pyruvate, every student, please listen. Whenever pyruvate converted into phosphoenol pyruvate, what is the enzyme involved here? Pyruvate phosphodikinase enzyme. It's a kinase enzyme. You know, friends, in order to convert pyruvate into phosphoenol pyruvate, there is a utilization of 4 ATP. There is a utilization of 4 ATP. That is for one cycle. But how many times it is recycling? 3 times. So 4 into 3, 12 ATP here. My friends, no need to get confused. I'm talking here. You know, I'm talking here. Where I'm talking, this one I'm talking. The pyruvate transported back to the mesophyll cell and it is converting back to the phosphoenol pyruvate. Every time it uses a 4 ATP. How many times it is turning the cycle? 3 times. So every time 4 ATP, 3 times 12 ATP. And here, one carbon dioxide taken by the Rubisco continue the Kelvin cycle. Here, Kelvin cycle, how many ATP we used? 18 ATP we used. So 18 plus 12, 30 ATP. So my friends, the question is crystal clear. In case of a C4 plants, in order to produce 1 glucose molecule, we require 30 ATP molecules, 6 carbon dioxide plus 12 NADH. There's no difference. When it comes to the C3 plants, for 1 glucose, we require 18 ATP, 6 carbon dioxide and 12 NADH. Is there any confusion now? Now you can ask me. When one pyruvate converted into phosphoenol pyruvate, there is a utilization of 3 ATP. Sorry, 4 ATP. So cycle occurs 3 times. We are spending 12 ATP extra there. So that's why in the C4 pathway 30 ATP, C3 pathway there is only 18 ATP. Clear, everyone? If you have any doubt, now you can ask me. Is it clear for so far? No. Right. Swati, that's correct here. One pyruvate is 2 ATP. Right. It's a dikinase enzyme. Any confusion, I will re-explain. Okay. All right. Okay. That is good. Okay. No. Here, there is no much partial pressure here like hemoglobin. That is entirely different because plant is completely different than human. No, because in case of our heart, lungs, partial pressure is exclusively high when compared to the plant tissues. So here partial pressure is not the greater parameter. Okay. I don't know how many people are there now in the class, but you remember my statements. If you are really with me till 50 days like this only, like this, trust me, I will help you in every point of your preparation. Okay. Just be dedicated till the end and don't think about result. Okay. If you keep on thinking about your result, you will always, worried. You just focus on your preparation. Okay. Now in the tomorrow class, we will continue the plant physiology of photosynthesis and nitrogen metabolism. Both I will complete. Okay. Now listen. Now the most important step. By tomorrow, before you attend 7 p.m. class, what is your responsibility? This is utmost responsibility. Now by early morning before 9 a.m., I will send the MCQ questions. only on photosynthesis, not on any other topic, only on photosynthesis. Now your responsibility is that you have to solve that MCQs. You no need to tell me also. You no need to tell me that you're solving it or not. You have to, you ask me if you have any doubt. So if there are 50 MCQs, I'm requesting you to solve at least 40 MCQs. Then that would become a good preparation. Okay. So by before 9 a.m., I will share the both recordings of today class and MCQs. If you solve MCQs, I believe you are good. Okay. All right. All right. Okay. I'll see you in the tomorrow evening class. SSV, I can't give the schedule, but I will give you the name of the chapters. Okay. Because I will take Sunday also more number of hours, especially. Okay. Sunday, I will take more number of hours. Okay. I'll give you the name of the units, but not exact topics. Okay. Okay. All right. All right. I'll see you in the next class. Okay. Thank you, all of you. Thank you.